Okay. Okay, so last week in our last lecture, I started out and I gave you guys a little bit of a reminder of what lipids are. And lipids can be divided into a variety of different groups, but essentially they are just something characterized by long carbon chains in some way, usually in a variety of different ways. The most common ones that we talk about often be things like fatty acids that will just be single chains of lipids, not to be confused with the steroids, which were those that has that sterile nucleus, which is the four rings together. But we're not covering steroids in this lecture. What we're covering is the beta oxidation of the fatty acids, which is going to be characterized by things like this here, this long carbon chain. And something which I think is really important that was going to help is that this here is this kind of carbon chain. We describe this as being saturated. And it's saturated because it is considered saturated with hydrogen. And this would be a saturated fatty acid if it had a fatty acid group at the end. But it can be unsaturated if we were to, let me see, where's the erase? There it is. It'll be unsaturated if we were to remove some of those hydrogens and put in a double bond. So it's considered saturated if it's saturated with hydrogen. If you remove some of that hydrogen, then it's no longer saturated. It'll become unsaturated. And what replaces the hydrogen is a double bond. And if you have one double bond, then it is mono unsaturated. And if it has several double bonds, such as, for instance, how am I going to do this? For instance, something like this, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, something like this. This would be poly unsaturated. I don't think this would exist in reality because the conjugation, but something like this would be poly unsaturated because there is more than one double bond. That's going to come up a little bit important later when we talk about these metabolizing these fatty acids, because it's a different pathway between the saturated, the mono, and the poly unsaturated fatty acids. Now we'll study the saturated first of all, and then we'll move to the mono and poly, which is just a little bit to be added on top. We're not going to be studying any of nothing like waxes, the phosphoglycerides and sphingolipids. We're not going to study those either, but it's worth mentioning on the side that the body has the choice of either metabolizing fatty acids to use to make energy, or it can use them to build membrane lipids. And so at some point, the body's going to say, well, what do I want to do? Do I need to build membrane lipids, such as phosphoglycerides, sphingolipids? Do I want to build them? Or do I want to burn the fatty acids for energy? And there's a switch, the switch between one or the other. Exactly the same as we were looking at with glycolysis and gluconeogenesis. There's a switch to say, utilize sugar and go down glycolysis. And there's another switch to say, use these amino acid intermediates, use these TCA cycle intermediates, use pyruvate, and go upwards through gluconeogenesis to build sugar, to build sugar and glycogen for storage. So sometimes your body wants to burn things, and sometimes your body wants to use things, and your body will burn things when it has and under different conditions. That's going to come up a little bit more. But for instance, when you've just eaten a big meal and you've got an insulin spike, your body's going to utilize sugar. But perhaps under starvation conditions, your body's going to utilize fatty acids. And under fed conditions, your body's going to use fatty acids to build things. And under starvation conditions, your body's going to use fatty acids to as an energy source. So that's an important thing to note. But in general, we're looking at, in this lecture, we're looking at burning fatty acids, and particularly where they've come from, the triglycerides, which is how your body stores fatty acids in your body, with that tri-acyl glyceride, where you have the glycerol molecule, which has got 3 OHs on it. It's just a carbon, 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 3 OHs, and then it will sterify with the fatty acid 3 times, 1, 2, 3, and join on, and you will get esterification, and you get this, which is I think you get this, which is the ester, and if you do it with the glycerol and the fatty acid, and this bond is often referred to as an acyl. So it is triglyceride or tri-acyl glyceride. It's just 3 fatty acids bound to a glycerol molecule by an ester or acyl bond. And this is how the body stores fat. This is how the body stores fatty acids, and it's a store of energy in the body. Also provides insulation and things like that. But this is all stuff that I kind of talked about last time. I'm just kind of breezing over a little bit to refresh your memory. So here we go. Oh, let's get rid of all of that. Okay, so I talked about all of this last time, and this isn't really super relevant. Okay, so I did mention saturated and unsaturated. I mentioned that the hydrocarbon chains are usually 4 to 36 carbons long, almost always an even number, and usually unbranched. In Biochemistry 1, it's always an even number, never branched. In Biochemistry 2, I'm going to give you guys an example when it's an odd number and when it does branch, but we don't talk about it very much. I just kind of put it out there and say that the process is slightly different, but just kind of leave it there. But that is introduced. I introduced how we named them, about the different omega fatty acids. I gave you guys some naming practice questions to go through. This won't really come up in the exam directly, but knowing this stuff will be important. And then this stuff is derived from the diet. And linoleic acid, for instance, is essential. So it's dietary essential. You need to get it from your diet. Your body cannot make it, but your body can make the other ones, can make the other fatty acids from that. I'm going to keep skipping forward. And what is beta oxidation? Well, if we look at the carboxylic acid, and it's going to be a chain that comes out, and the carbon number 1 is the carboxylic acid carbon, the carbonyl carbon, but the carbon next to it is often referred to the alpha carbon, and the carbon after that will be beta, and obviously the carbon after that is going to be gamma. And beta oxidation is if you have this fatty acid, it's an oxidation at the beta position. And so it's just really, we're going to cover that today. There are 4 steps to how this this carbon at the beta position gets oxidized. And there's a question for you guys. So if we're going to oxidize this carbon here, the beta position, what do you think we're introducing to that carbon?
An alcohol first?
Yeah, oxygen in general, but yes, an alcohol first. So there's going to be the first step is we're going to introduce a double bond. Then we're going to hydrate that double bond. And then we're going to oxidize that double bond. And then we are going to, that creates, you now have a, oh, there's my cat. We're going to, we now have a carbonyl here. We now have a carbonyl here. And so that creates a lot of kind of strain on this this molecule. And it makes it so that it's really ripe for this area here to be cleaved. And so it will cleave it in the final step. So the first step is the introduction of a double bond. The second step is to hydrate that double bond to an alcohol. The third step is to oxidize that to a carbonyl. And then the fourth step is to cleave between the alpha and the beta position. And this, and so then you have this 1, 2, carbon comes off. And as part of the cleavage process, you get a coenzyme A attached onto here. So if we have a coenzyme A with an acyl group, what do we call that one? That'll be
acetyl-CoA.
Acetyl-CoA. Yes. And so what can we then send acetyl-CoA to?
The TCA cycle.
That's it. Then we can feed that into the TCA cycle. But then what we've now done is that we've now made our carbon chain 2 carbons shorter. We've introduced that carbonyl here, and as part of the process, we'll get an alcohol here. And so now we've got we've now made it 2 carbons shorter. We've got a fresh carboxylic acid to do it all over again. And you just keep doing that sequentially again and again and again, just burning through. I'm going to go through those steps in detail later on, but I wanted to frame it to give you guys an idea of what's going on. It's just a sequential oxidation at the beta position, cleaving off those 2 carbons. When you cleave it off, you make that fatty acid 2 shorter, but then it still has that carboxylic acid group. And then you could just do it. You created a new beta position, and you can just keep doing that again and again and again until you run out of carbons. And so you're introducing acetyl-CoA into the system that can be utilized for food, that can be utilized for energy by putting it into the TCA cycle and just oxidizing it repeatedly to take its electrons and feeding it into the electron transport chain. And what this should kind of give you guys a memory of a little bit is actually of the TCA cycle, because there is a step of the TCA cycle where you introduce a double bond, hydrate it, and then oxidize it. And that happens in the last 3 steps of the TCA cycle. And nature has a way of repeating that. Those first 3 steps of beta oxidation, the first 3 or 4 steps of beta oxidation, double bond, hydrate, oxidize, and then cleave. Those are the 4 steps. The first 3 steps are pretty much exactly the same as the final 3 steps of the TCA cycle. And it's that pattern that why reinvent the wheel? Nature has a habit of repeating the same chemical reactions in those biological processes, which is kind of cool. But this is going to come up in detail a lot more when I talk about it. I just wanted to introduce it now. So, okay, so I kind of framed the lecture last time. And I talked about how fats are stored in the body as lipid droplets, that they're taken up in the intestines. This is really just a biology slide that you can kind of look at if you're interested in. You won't be tested on this. But I will have a lightly test on lipoproteins. The idea that an alipoprotein is this down here, an alipoprotein is this protein. It's kind of like a belt that's made up of alpha helixes, which are really soluble. And of course, fat is not soluble. And so these wrap around the fat, kind of like shown here. They wrap around the fat. And because the fat doesn't want to dissolve in water, these wrap around it. And so they kind of create a hydrophilic coat over the lipid to get it to solubilize a little bit better. And that's alipoproteins are the name of the protein. And then when it binds to the lipid, it's a lipoprotein particle. And there are a bunch of these different lipoprotein particles, depending on their density and size, where if they are bigger, they tend to be less dense. If they are smaller, they tend to be more dense. And the high density ones are the ones that don't really transport triglycerides so much, but they do transport cholesterol more. whereas the low density, the slightly larger, low density lipoprotein particles are the ones that tend to transport the triglycerides. And that's kind of summarized here. I'm going to probably test very lightly on this kind of stuff. I would like you guys to be comfortable with the idea that lipids aren't soluble in water. It's not soluble in blood. How do we solve that? We wrap them in a protein coat, a lipoprotein protein coat, in order to transport them, to solubilize them. There's different ones for they're transporting different fats, and they're classified by their size and density. And perhaps the very low ones are the ones that transport triglycerides, and the high ones are the ones that transport cholesterol. And that is why it's important to, when the doctor tests your cholesterol, they usually don't test your cholesterol, but rather they will test your lipoprotein concentration, because there isn't much cholesterol in your blood. It's more that it's stuck, because it's not solubilized, it's stuck to a lipoprotein. It's not free. It's so we're more interested in the lipoprotein. That's getting a little bit more clinical. But anyway, I covered all of this. I'm going to skip forward a little bit. I don't want to get too stuck behind. So where I ended up with is this fatty acid acetyl-CoA synthase, and that is a set of enzymes which are present in the mitochondria. And when fatty acids are taken up by the body, right, let's change color. Let's change to a green. When fatty acids are taken up by the body here, they first need to be activated. They don't hang around as fatty acids too long. So this is where they're not being stored, but your body wants to use them for something. And so, when they're being stored as triglycerides, and then those triglycerides get released into the blood, cells take them up, and then they want to do something with them. And before they can do something with them, they need to activate them. And here is that fatty acid here. And I went over this last time, so I'm not going to go into detail, but it needs to be activated. And to activate it, you need to turn it into a fatty acid, fatty acid CoA, which is a fatty acyl CoA, because this is an acyl bond here. That kind of old-fashioned way of it's a thioester, but a lot of times an old-fashioned way of calling them is an acyl bond. And so I discussed last time that what happens is that first of all, the fatty acid needs to be activated by ATP, and so you have this A, P, 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 and the first thing is that the alcohol of the carboxylic acid will attack this phosphate, and it's the first phosphate. If we have 1, 2, 3, normally it attacks phosphate number 3 and becomes phosphorylated. This time it doesn't. This time it attacks phosphate number 1, the one that's closest to the adenosine base, and it will, or nucleoside, the adenosine nucleoside. It's not a base. A nucleoside is the base plus the sugar, and adenosine, because it means osine, that means that it is the nucleoside, which I cover in my Biochem 1. But normally it would attack the terminal one. This time it doesn't. It attacks the phosphate number 1, and that means that it will attack the phosphate number 1, and you'll get it. You can see here that. Here's the carboxylic acid with P adenosine, and it makes the fatty acyl adenylate. Adenylate is just the adenosine with 1 phosphate. That's adenylate here. And so it'll be the carboxylic acid adenylate, and that activates it, because the adenylate is a really good leaving group. It's a really good leaving group. So normally it would just be phosphorylated. Let me change color. Often it is just phosphorylated like that by picking up the terminal phosphate. This time it doesn't do that. This time instead it picks up this group and then becomes adenylated, and it makes it a really nice leaving group so that the acetyl-CoA, the sulfur and its lone pair of electrons can attack that that carbon. That carbon's going to be delta positive because it's got two oxygens attached to it, and because this is a really good leaving group, that's going to be able to get that bond is going to cleave. The bond's going to go back to the oxygen. It's going to cleave it, and you're going to generate the fatty acid CoA, and that acyl CoA. And the reason why we do that is that it needs to be adenylated first of all, because if you just had this fatty acid like this that has not been adenylated, and the acetyl-CoA, the CoA, the CoA tried to attack it, it can't attack it because it's not activated enough. It's this oxygen here on its own is not a good leaving group, because it would be like this. Let me switch to a nice green. If this SH here, that lone pair of electrons, acted as a nucleophile, attacks this carbon here, which is going to still be delta positive because it's got two oxygens next to it. This oxygen here, you can't just get this oxygen here coming off and leaving. It's an extremely poor leaving group. So you need to activate it first of all, and you activate it by adenylating it. Now, why it is adenylated, usually what happens is that it will get phosphorylated, and then that creates the good leaving group for the SH to come. But in fatty acyl CoA synthase, it gets adenylated instead. I'm not completely sure why that is. Well, actually, I'm fairly sure why that is. It will be because it's more energetically favorable to do so. It's more for some reason, it is more energetically favorable to be adenylated than to be phosphorylated. And it's just that nature and evolution just went down the path, and it turned out that it has two options: get phosphorylated, get adenylated, and the adenylation just created the more energetically favorable product. And so that's the one that ended up being chosen. The exact chemistry of why, I'm not completely sure, but it's because it'll be more energetically favorable. And then the final thing I want to say about this slide, just briefly, is that if you make the cleavage here, then this adenylate group is going to go on here, and that's what we have here. But then you get these two phosphates, this P, that's a P, and we give it I because it counts as inorganic phosphate, which is just something that biologists and biochemists do, is that when they get these phosphate groups, they call it inorganic phosphate. And where you have these two phosphates that have popped off, we call that pyrophosphate. So normally, or not normally, often in these types of activating reactions, a single phosphate comes off and you have ATP. A single phosphate comes off to phosphorylate and you get ADP plus a PI from the phosphorylated thing, whatever it is in this case. But this time it's the other way around, and you end up with AMP plus pyrophosphate at the end of it. I kind of mostly went over this in the last lecture, and I went over it just real kind of quick this time, just to refresh your memory, and I did build on it a little bit this time. So I believe I'm going to move on to something new. Yes, I'm going to move on to something new. Just to remind you guys, the reason why we do this is because the fatty acid, before we can do something with the fatty acid here, it needs to have the CoA added to it. And then once it has the CoA added to it, fatty acid CoA, we can then put it into catabolism or anabolism, but whichever one you choose, catabolism to burn it and to utilize it for fuel, or anabolism to use it to build other fatty acids or membrane lipids or things like that. Whichever one you choose, doesn't really, in either case, it always needs to be have the CoA added to it pretty soon after entering into the cell in order to activate it to make it ready to go into either pathway. But I'm going to talk about, I'm not going to talk about the anabolic pathway. I'm going to now start talking about the catabolic pathway. Before I do, does anyone have any questions about fatty acid acyl CoA synthase? Bearing in mind that I did go through it a little quick, but I did that because I did go through it more last lecture. Okay, I'm going to move on then. So what happens next is that once you've had the acyl CoA added to it, the body then needs to make two choices. It is either going to burn it or in a catabolism or build something with it with anabolism. In the case of catabolism, it then is going to get transported into the mitochondrial matrix. At this point here, it is in the mitochondrial, it's on the mitochondrial membrane, the outer membrane, but you then need to transport it into the mitochondrial membrane, into the matrix, because the matrix is where it gets burned by beta oxidation. And thus what you're doing actually is that because there's a membrane between the matrix and the, well, outside the matrix, the intermembrane space in the cytoplasm, there's a membrane. And so what you're effectively doing is, and things can't pass through a membrane easily. Now there's a special transporter to get the fatty acid CoA in, but not one to get it out. So what we're doing is we're building two pockets. We're building a pocket inside the matrix where we specifically pick and choose fatty acids to go into the matrix to be burned. And then once they enter into the matrix, that's it. They are committed to being burned by beta oxidation. But if it isn't in the matrix, it's outside the matrix, it's in the cytoplasm or the IMS, that is where it's saying, well, I could be used for either. Maybe I'm going to be used for anabolism. Maybe I'm going to be used for catabolism. But once it gets transported into the matrix, there's no getting out and it's going to get burned. And so there's a specific transporter to get it in, but there's never anything to get it out. And the transporter to get it in is the carnitine shuttle. And I'm going to talk about the carnitine shuttle, and I'm going to ask fairly deeply on the carnitine shuttle. Nai, you have a question?
Yes. So with the beta oxidation, you're saying that it only happens in the matrix, or does it also happen in the mitochondria?
The matrix is in the mitochondria.
Oh, sorry.
The matrix is the innermost part of the mitochondria. It's
Oh, okay.
outer membrane, IMS, inner membrane, matrix.
Oh, so it's just happening deeper. Okay. Got it.
Yeah. So this is the structure of carnitine. And really, in a roundabout way, it's kind of an amino acid. It's not, but it's related to the amino acids. It helps transport fatty acids through the mitochondrial membranes into the mitochondrial matrix. And really, essentially, it's kind of just a tag. It's kind of really that you have if this is the membrane here, that red line, and here's a transporter, and this transporter, and I'm just going to draw it very oversimplified. There's two membranes, there's an IMS, but I'm just going to go cytoplasm and matrix. There's more in between. I'm just oversimplifying. And this really just picks up anything that has a carnitine attached to it and transports it into the matrix. And so if we tag a fatty acid CoA with carnitine, then the carnitine transporter will grab it and pull it into the matrix. And it just so turns out that fatty acids are the only thing that get carnitine added to it. And so when you get a fatty acid and you get a carnitine to it, you're committing it to getting transported into the matrix to get burnt. If you don't put a carnitine on it, it will stay outside the matrix in the cytoplasm or the IMS and could be used for something else, such as anabolism and building membrane lipids. But once you add a carnitine to it, the carnitine transporter is going to grab it and pull it into the matrix. And the shuttle is here. So what happens is that I'll run through it. What happens is that you have the cytosol, the IMS, and the matrix, like I was saying, the outer mitochondrial membrane, the inner mitochondrial membrane. And what happens is that you might have some fatty acid, CoA, floating around in the cytosol. It is uncommitted. Maybe it will be anabolism, maybe it will be catabolism. A carnitine gets added to it, and the CoA is exchanged. So it's notice carnitine, the enzyme is carnitine acyl transferase. Think about it. All of these things, they all tell you what they do. Transferase, transfers. So the acyl CoA will come off and get replaced with carnitine. I've drawn it slightly wrong there. Will get replaced with carnitine to generate the carnitine ester instead. So, and the CoA comes off. I'll run through that again. So you get an exchange. You got a fatty acid CoA. Carnitine comes in and replaces the CoA, which comes off and re-enters the pool of CoA to be used for something else. So the coenzyme A pops on and the carnitine pops off. Right, sorry, the CoA pops off and the carnitine pops on. They exchange, and then that means that the CoA is now free to do something else. And this fatty acid here has the carnitine on it. And like I said, the carnitine transporter is now going to pull it through into the matrix. And you now have a fatty acid carnitine in the matrix. And now you just do the reverse. An acyl transferase now takes off the carnitine and frees it. And a CoA that is present in the mitochondrial matrix attaches to it, generating fatty acid CoA. And so you have a pool of fatty acid CoA in the cytosol that could go either direction. And you have a pool of fatty acid CoA in the mitochondrial matrix, which is only for catabolism. And you have a pool of CoA, acetyl CoA in the, no, CoA, just CoA, not in the cytosol, and a pool of CoA in the matrix. And they're all separated. They're all separated by lipid membranes, which is creating different environments, and you're separating out your chemical reactions. It's exactly like being in a chemical synthesis lab, and you've got two beakers, and you have two chemical reactions in those two different beakers, and you've got some fatty acid and CoA in one, some fatty acid and CoA in the other, and they are two separate compartments. And you can, but you could use carnitine to transport between those two different beakers. It's exactly the same kind of thing. We have lipids creating lipid membranes creating two different compartments, where you have a compartment in the cytosol of fatty acid CoA, and carnitine, which may exchange in different ways, and can either be used for anabolism or catabolism. And you have a pool of fatty acid CoA, carnitine, fatty acid in the matrix. And they're used for different things. That's only going to be used for catabolism, but that's separated. And that means that your body doesn't put in all of the effort to start burning a fatty acid and be like, oh no, actually, no, I've changed my mind. I want to build something instead. It's so that you can't do anabolism and catabolism at the same time, because you'll just get a futile cycle. By separating them into two different compartments, you'll never do anabolism and catabolism at the same time, and you won't get a futile cycle, and you won't waste energy. I'm going to go into a little bit more detail about this in a moment, but Lauren, you have a question?
Yeah, I just wanted to make sure I understand. So, the carnitine acetyltransferase 2, does it trap the the carnitine group that came in by removing the carnitine and replacing it with the CoA? So it traps it
Uh,
in the matrix? I don't know if that makes sense.
so yeah, good question. So I was just about to get to that, but what happens is that the well, let me skip forward a little bit. Let me go and do the rest of it in sequence. So there we go. So the first step is that, like I said, that the CAT1, carnitine acetyltransferase 1, exchanges a trans esterification. So trans, that means to exchange. Esterification, you're going from a thioester to a carnitine ester. Um, to transiently attach fatty acids. Oh yeah, fatty acid CoA to the hydroxyl group, the alcohol of carnitine. So what we're doing first of all is we're exchanging for the fatty acid, exchanging the CoA for a carnitine to generate the carnitine fatty acid. So that's step one. Step two is that this is out of order. Step two is that the transporter will now pull this through, and it's now in the mitochondrial matrix. And then step three is that in the matrix, CAT2 will do the reverse of CAT1 and exchange the carnitine for the CoA. And so that'll mean that you'll use up CoA in the matrix and generate carnitine. But what happens is that there's this transporter here. This transporter here is as an antiporter, and what that means is that something goes in one way and something else goes in the other way. And what goes in one way is the fatty acid carnitine that goes in one way, and in return, what goes in at the same time, what goes in the opposite direction is the released carnitine from the matrix goes back into the cytosol. That's called an antiporter. And so a lot of transporters require energy and ATP. By doing a swap, it doesn't require energy or ATP. And so you at this here, you swap a carnitine fatty acid going in for a carnitine on its own going out to replace the carnitine that you use by generating the fatty acid carnitine in the cytosol. Does that answer your question?
Yes, thank you.
Um, I'm going to very briefly, I'm going to skip forward a little bit because I want to make sure that we cover the 4 steps of the fatty acid degradation. So I'm going to go a little fast for about 5 slides, but there's not too much detail on these 5 slides. One thing that I did want to mention is that this step 1 is CAT1. And so step 1 is going to be the one that you want to regulate, right? If you're in a state where you want to build or you're a state you want to burn, then you're going to want to regulate step 1. And this is kind of like step 1 of fatty acid beta oxidation, in that it's not the part of the beta oxidation, but it's like a pre-step. This kind of pre-step 1. And so you're going to want, if you don't want to burn fatty acids, you want to burn right now, then the best one to inhibit is this very, very, very first step. And you can keep your fatty acid in the cytosol to build. And so malonyl CoA is the first metabolite, the first thing that's made when you're building fatty acids. When you have an excess of sugar in your body will turn that sugar into fat, and you're in a state of anabolism. Because you have a lot of sugar in your body, you're in a fed state, and you're going to go into, you're going to want to store some of it, and that's going to be in a state of, that's going to be an anabolic state in terms of the fatty acids. If you eat lots of sugar, catabolic sugar, anabolic of fatty acids. So you're going to turn some of that sugar into fat. And the first step of turning sugar into fat is turn it into malonyl CoA. And so if you have a large amount of malonyl CoA, that is signaling we're in a building stage. We're in a fat building stage. And you don't want to you don't want to burn your fat if you're in the middle of building your fat. You want to store your fat until you need to burn it. You don't want to burn it when you're in the middle of building it. So where you have a high concentration of malonyl CoA, that's signaling you're in a fat building stage. And so your fat, this malonyl CoA will inhibit carnitine acetyltransferase 1. And it won't inhibit it as being in a normal inhibitor. It will act as an allosteric negative modulator. Who can tell me what an allosteric negative modulator is? Anyone?
Isn't it when something binds outside of the active site and it changes the positively or negatively or positively changes the activity of the enzyme or something like that?
Yeah, and since it's negative, it will decrease the activity. So malonyl CoA is the first step of turning sugar into fat, signaling that your body is trying to turn sugar into fat, signaling that we don't want to burn fat right now, and so let's not burn fat right now. So let's inhibit the step of taking fat, putting it into the mitochondria to burn it and utilize it. And so it will act as a allosteric negative modulator and inhibit this enzyme. That little symbol there means inhibit enzyme. So we'll inhibit that enzyme. And so it will keep all the fatty acid CoA in the cytosol, so it can be continued to be used to build bigger and longer fatty acids or membrane lipids and things like that. Next slide, I think I'm going to skip over kind of quick. This is just another illustration showing what I've already told you guys about. I'm not going to talk about that. I've already told you guys. Oh, and just very briefly, is that the majority of the when you burn triacyl glycerides, this is the glycerol, and you have a fatty acid attached here, here, and here by that acyl bond, that ester bond. And so the majority of the energy comes from the beta oxidation of fatty acids. But if you really need to, you can take glycerol and put it into the TCA, into glycolysis, and then it will turn to pyruvate in the TCA cycle. So you can, if you want, you can also burn the glycerol of triacyl glycerides and use them for energy, and they can just go straight into glycolysis. But the majority of the energy is in the long chain fatty acids attached. I just wanted to very briefly mention that on the side. So now we are gonna switch gears. If I can make it move to the next slide. Okay, I'm going to switch gears and talk about the process. I'm going to skip over the next 3 slides real quick because there's not too much to say here. I've already kind of told you guys this, is that what we do is if we have our fatty acid here and it's beginning here, carbon 1, 2, 3. Let me change color. Carbon 1, 2, 3, 4, 5, 6, etc., etc. That this carbon here is the carbon number 1. So this one here is the alpha carbon. This carbon here is the beta carbon. And like I was saying, you at step 4 of beta oxidation, you cleave between carbon alpha and beta, carbon 2 and 3. You cleave here, and in doing so, you at the same time as the cleavage, you get a CoA adds on to here. And so you have acyl acetyl CoA, and then that enters into the TCA cycle, and your fatty acid is now 2 carbons shorter. But as part of that process, this will also be a carboxylic acid now. When it gets cleaved, the CoA goes onto 1 and 2, and the fatty acid group is put onto the beta carbon, and that becomes a fresh 1, 2, 3, 4 with a fresh alpha beta and a fresh cleavage between them when you do another round of fatty acid oxidation. So you just keep doing it in rounds of cleaving off carbon 1 and 2, creating a new carbon 1 and 2, and then cleaving that off again. and then creating a new carbon 1 and 2 and cleaving that off again and doing it again and again and again in pairs until you run out of carbons. And so now you kind of see why it's usually even, because if it was odd, then you'd be stuck with 3 carbons at the end. But that's why it's always even, because you remove 2 at a time during beta oxidation. And so it's kind of summarized here. Undergo oxidative removal of successful 2 carbon units in the form of acetyl CoA. This is a perfect definition. Fatty acids undergo oxidative removal of successive 2 carbon units in the form of acetyl CoA. That's a really nice way of doing it. So if you have something like palmitic acid, which is 16 carbons long, going to turn it into 18 acetyl CoAs. And then those CoAs are going to enter into the citric acid cycle, TCA cycle, to make NAD and FADH2, which is then going to go into the electron transport chain. Yeah, and we covered that in detail, so I don't need to tell you guys about that. But it's just really kind of like glycolysis. Glycolysis, sugar feeds into the TCA cycle to generate NAD and FADH2, which then feeds into the electron transport chain. And it's that same thing. Nature recycles chemical reactions. Catabolic processes converge. Everything just converges into making acetyl-CoA. Sugar, amino acids, fatty acids, they all just make acetyl-CoA, so they could just go into the citric acid cycle, make NAD and FADH2, and then go into the electron transport chain. So what are those 4 steps of fatty acid oxidation? I really want to finish this this lecture, but this is really actually the meat of the lecture here. This is how we do it with saturated fatty acids. We're not going to get to do mono unsaturated and polyunsaturated in this lecture, but I want to get to the end of these 4 steps in the next 10 minutes for this lecture. And then we'll cover the rest in the next lecture. But essentially, I'm going to show you step 1, 2, 3, and 4 in detail. I gave you guys a little bit of an introduction at the beginning as well. I'm going to run through the steps now briefly and then do them in detail. Step 1, we start out with our fatty acid CoA. Here it is. There's the acid CoA, and here's the fatty bit. The first step is a dehydrogenase. Dehydrogenase enzymes typically turn single bonds into double bonds. But what happens is that a single bond turns into a double bond. But what you've done there is that you've taken off 2 hydrogens. And where you have those 2 hydrogens, that's a bond. So there's electrons. And so you've got this. You've changed the number of electrons. You've got. But in this system here, you've got 1, 2, 3, 4, 5, 6 electrons. But in a double bond, you've got 1, 2, 3, 4. So a dehydrogenase is where you go from a carbon-carbon single bond to a carbon-carbon double bond. But what you're doing there is the carbon has lost electrons. What do we call it when a carbon loses electrons? Or anything loses? Sorry? Other way around. Oxidation.
Guys.
Oxidation is loss. So the carbon becomes oxidized. And so if carbon becomes oxidized, something's become oxidized. Oxidation is coupled to reduction. So in step 1, it's a dehydrogenase, which tells you go from a carbon-carbon bond to a carbon-carbon double bond, losing hydrogen, but also losing electrons. And those electrons, so that's oxidation. Oxidations are coupled to reductions. So in this case, you actually turn an FAD into an FADH2. which is really nice. So actually, not only from beta oxidation do we generate acetyl-CoA that enters into the TCA cycle, but you also, in step 1, you generate an FAD on a side. That's super handy. And so you generate it and you put in a double bond here. And then what happens is that double bond gets hydrated here. That's just a simple hydration, nothing else to it. Hydratase, a hydratase hydrates it. Third step, like I say, there's an oxidation. This alcohol is oxidized to a carbonyl. And because something is being oxidized, nothing happens here. This is just a hydration. It's not a redox reaction. But this step here is an oxidation. So again, something gets oxidized. You go from an alcohol to a carbonyl, which is more oxidized. So that carbon's been oxidized. It loses electrons. And what gains electrons? NAD to NADH. And then the final step, you get a cleavage between the carbon 1, 2, 3, carbon alpha, carbon beta. You get a cleavage between the alpha and the beta. So carbon 1 and 2 go off on one side. Carbon 3, 4, 5, 6, 7, 8, 9, 10, whatever, go off on the other side. And so this acyl CoA comes off. And at the same time, a CoA goes on to here, onto here. Let me change color so you can see. The CoA goes on to, there you go. This CoA goes on to here. And now you have a fresh fatty acid, fatty acid CoA, and an acetyl CoA comes off. And the enzyme that does that is a thiolase. Thio means sulfur, lase means to cut, saying that we are cutting by using a thiol, which is the SH of acetyl-CoA. And remember, the acetyl-CoA is just CoA, which is a big fat molecule with an SH added to it. So the SH comes and attacks here, generating the thioester, thiol CoA, and in doing so causes a cleavage that causes the acetyl CoA to come on. So I'll run through these steps in the next 5 minutes, a little fast, but it's not too bad. I've explained it, so it's not too bad. So the reason being, actually, the logic is that the logic is that this is a carbon-carbon bond, and you've just got a carbon-carbon bond with HH. And so this is what's often referred to as a methylene group. A methylene group is just a standard CH2. So you got CH2, CH2, and that's super stable. That is super stable. This bond right here is super stable. So it's very hard to cleave. What do? We turn it into a double bond, we hydrate it, we turn it into a carbonyl bond, and now instead it's the carbonyl, and that is much easier to cleave. That's why we do it, because this is hard to cleave, but this, where it's a carbonyl instead, is much easier to cleave. That's the logic. That's why we do it. So, and that's kind of explained on this side here. And like I was saying, step 1, you have the alpha carbon, and between the alpha and the beta carbon, and between the alpha and beta carbon, you get a double bond added. And this is very similar to the step 6 of the TCA cycle, actually. But succinate dehydrogenase, remember succinate dehydrogenase? This is actually doing the same thing as succinate dehydrogenase does. I remember what succinate dehydrogenase does in the TCA cycle. Succinate dehydrogenase is right here. And you have succinate to fumarate, where you turn a carbon-carbon single bond in succinate into a carbon-carbon double bond for fumarate, pulling those electrons off, feeding them into FAD, and then feeding them into Q for oxphos. It does exactly the same thing here, where the fatty acid is turned into the enol fatty acid, which is that double bond. And then the electrons from that are fed into FAD, which feed into the TCA, which feeds into Q for the electron transport chain. So it's actually complex 2, does it with sugar, and these enzymes here, the reductases and the step 1 of the beta oxidation, they do essentially the same thing. They turn, but with fats, they turn a single carbon-carbon bond into a carbon-carbon double bond, oxidizing it, putting it onto FAD, putting it onto Q, which is for the electron transport chain. Does the same thing. Nature just reuses the same process. Not exactly the same enzyme, but it's a very, very similar process that does a very, very similar thing. So step 1, I'm going to go backwards a slide. Step 1 is that double bond between the alpha and the beta carbon. I'm going to test pretty hard on step 1, 2, 3, 4. Step 1, the double bond between alpha and beta carbon, oxidizing the carbon, reducing FAD to FADH2. The enzyme is this trans, so it's introducing a trans bond. Delta 2, indicating that it's the double bond starts at carbon number 1, 2, 3, carbon 2. Double bond starts at 2. That's what delta 2 means. Delta in naming means 2. And enol is just that this is an enol type bond here. And that's the enzyme. Sorry, that's the molecule, and this is the enzyme, the dehydrogenase. Skip past this, skip past this. Like I said, it's just like succinate to fumarate. Just like it. Very similar. Step 2 is the hydrogenation, just like fumarase in the TCA cycle. Just like fumarase in the TCA cycle, you get that which just after you generate the fumarate, you then hydrate the fumarate in the TCA cycle, and exactly the same thing. You have this double bond, and you hydrate it using this hydratase. And just like fumarase, you have a enol hydratase. That's step 2. Step 3 is it gets oxidized again by dehydrogenase, which is again coupled to the reduction of NAD to NADH, which then enters into the electron transport chain using a dehydrogenase again. Similarly, this is the same as the malate dehydrogenase of the citric acid cycle. And just like that as well, it's just specific for the L stereoisomer as well, because it's you started out with the trans double bond, which is going to generate when it hydrated, it's going to generate the L form of the fatty acid, and so only the L form of the fatty acid is going to get reduced to oxidized to the carbonyl, generating turning NAD into NADH, reducing NAD to NADH, creating a ketyl acetyl group, because this is now a ketone. And then the final step is cleavage by the thiolase, and you have this carbonyl here. That makes it. If it was the methylene group, then it'd be hard. But because it's these two carbonyls, especially this carbonyl here, you can get a cleavage here, and you then cleave this off, and this is acetyl CoA, and an acetyl CoA, and then a CoA can come in, and the SH is going to attack this carbon here because it's slightly delta positive. And you have the lone pair of electrons on this sulfur. It's going to come in, attack this carbon here. It's a reverse Clason rearrangement because essentially what's happening is that lone pair of electrons attack here. At the same time, this will go like this, and you get a cleavage between them, generating fatty acid CoA, 2 carbons shorter, and acetyl CoA. I think that's it. We can cover the next in the next lecture, but that's it for today. I'm going to finish up fatty acids in the next lecture, and then hopefully start on amino acids in the next lecture. We're getting to the end of the course. Amino acids oxidation will be the final thing. I'm going to set a quiz tomorrow on what I've covered today, and I'm probably going to set the homework as well. And you should be able to do the last question of the homework. You may not be able to do for 2 days until we cover ketones, but in the next lecture. But you can do like the first 9 questions. I'm going to set the homework tomorrow. You should be able to do the first 9 questions, and you can do the 10th and 11th question on Thursday when I finish the material. And I'm going to stick this video up on YouTube, and I'll send an announcement in an hour once it's uploaded to YouTube. But thank you very much, guys, and I'll see you in a couple of days. Have a good rest of your day. Oh, and if anyone has any questions, I can take any questions right now as well before I shut everything down, but feel free to go.
So with the last step, you're saying that the acetyl-CoA that has the SH will attack the carbon with the most carbons already on it to make fatty acid CoA and then regular acetyl-CoA?
Yeah, so what happens is that a CoA, which is present, that sulfur will come in and attack the beta carbon. It's carbon 1, 2, 3, which is alpha beta, will come in and attack the beta carbon. And because it's slightly positive because of the carbonyl, and then when it attacks that one, you also get this bond breaking. It's not going to attack this carbon because it's already got an acyl CoA on it.
Okay.
It attacks the beta carbon. It causes a cleavage between the alpha and the beta carbons, which are carbons 2 and 3.
Okay. And then the only difference is once it's cleaved is that one set only has 2 carbons to make it acetyl-CoA, and then the other set has multiple carbons to make it what did you say, fatty acid CoA?
Yeah. So you start out, for instance, with an 18 carbon fatty acid CoA, and then what you end up with is a 16 carbon CoA plus acetyl
okay.
CoA, which is 2 carbons.
Okay, that makes sense.
And it's kind of similar. You can kind of see it in this slide here, kind of like this. You just keep shortening it, going down, shortening it, and taking off 2 carbons each time.
Okay.
As acetyl CoA.
Okay, that makes sense.
Okay.
Okay, thank you.
Any questions? Yeah, no problem. Glad to help tonight. Okay. I'll call it that then. All right. Thanks very much, guys. See you.